Hello, dear students. I welcome you all on behalf of Shri Jaindra Rank Zone classes. Dear student, we are in a continuation of the lecture series of a class 11 chapter, that is anatomy of flowering plant. In previous class, we have talked or we have discussed about the tissues and the type of tissue, in which we have discussed already the different type of meristem regions where meristematic tissue are located. And we have also discovered the simple and compound permanent tissues. Now, these all tissues, how they are arranged in plant, that is the one of the important point. And when we see in plant, it is known that in plant they are arranged and forming a group of tissues that is known as tissue system. So our today's topic is tissue systems. So first is, what is the tissue system? So we have discussed the types of tissue based on the type of cells present in the previous few classes. But let's just now consider how tissues vary depending on location in the plant body. How the different type of tissues vary based on their location. So on the basis of their structure and location, what is the type of structure they have and where they are located. There are three type of tissue systems, which includes the group of tissues. And in this three type of tissue system, we are going to discuss the epidermal tissue, which will include the outermost layer, that is epidermis, and the structures which are associated with this. Then we will discuss the second ground or fundamental tissue system, and then we will talk about the vascular or conducting tissue system. Okay, so these three we are going to discuss in detail one by one. Okay, so I hope you understood what are this tissue system, where the location and the structure we are going to discuss. When we talk about the first tissue system, that is the epidermal tissue system, dear student, here it is going to include the epidermis and the structures or components which are associated with the epidermis. And we know that epidermis is the outermost covering of whole plant body. So it is epidermal tissue system is going to form the outermost covering of the whole plant body. And when we talk about this epidermal tissue system, it consists of epidermal cells. They also have stomata in the epidermis. There are some epidermal appendages like trichome and hair. So we are going to mainly talk about these three things. That is what epidermal appendages, okay, which include trichome and hairs, whereas the epidermal tissue system has the stomata. Stomata is considered as epidermal structure. It is considered as epidermal structure. Okay. Now, the epidermis is the outermost layer of the primary plant body. And you can see here, when we talk about the outermost layer, you can see this layer is representing the epidermis. And in this epidermis, you will find this structure which is known as stomata. Okay, you will see the presence of this structure, that is stomata here. Okay, then you will see the upper epidermis, lower epidermis. There is a cuticle which is present on the outer side of the epidermis. Thus, all structures are associated with this. So, if you talk about the epidermis, it is made up of some of the elongated and compactly arranged cell, and they are forming a continuous layer. So it is forming a protective sheath. It is forming a continuous layer over the whole plant body. Now, the epidermis is usually single layered, and the cells of epidermis are parenchymatous with a small amount of cytoplasm lining the cell wall, and they have a large vacuole. One of the important points, see, These epidermal cells are made of how much? They are made of elongated some cells, but they are closely packed. Means intercellular spaces are not going to present. But when we see one cell, within one cell, you will find a large vacuole. Suppose this is a cell, then this have a large vacuole. And when there is a presence of large vacuole, the cytoplasm is going to be peripheral. So this is a single layer of epidermis as we have discussed. And you can see here they are made up of elongated strands, they are closely packed, and they have a large central vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle. On the outer side of epidermis, you will find a presence of cuticle, which is a thick waxy covering, and it prevents the loss of water. Now, this cuticle, if we talk, then it is present on the stem and the leaf mainly, but it is found to be absent in the roots. Why? Because roots are going to absorb the water. If cuticle is impermeable to water, and if it is present on the root, it will not allow the root to absorb the water. Because of this, cuticle is absent in roots. Then you will see the presence of cuticle like this. The upper epidermis is also covered by a cuticle, as you can see, and the lower epidermis is also covered by the cuticle. So on the both the side, upper and lower epidermis are covered by a cuticle, which are impermeable to water. So it will not allow the leaf to carry out the loss of water. And because of this, they are present on the absent in the root. Now, this is the water which is lost from the epidermis. You can see, and the stomata are the structures present in the epidermis of leaves. When we talk about this stomata, this is present in epidermis, then it is a stomatal structure. And this stomatal structure, you can see, having a particular regulate the process of transpiration and gaseous exchange. It also carry out the loss of water in the form of vapor and also carry out the gaseous exchange. So the stomata are here in the epidermis. When they are turgid, the stomata get open. In the stomata, the guard cells, this particular two cells which are shown here, kidney shaped, they are known as guard cells. When the guard cells have a watery content, water rich in water, they are going to swell, and they will swell. The swollen guard cell are going to be turgid, known as turgid guard cell. And when the guard cell get turgid, the stomata get open. But when the guard cells become flaccid, the stomata get closed. Okay, so this is the thing which is observed in. And when guard cell is going to be flaccid or a shrink, when the water loss occur, this lead to the closing of the stomata. As you can see water is going outside and they are getting closed as the shown in the diagram. So each stoma is composed of two bean cells. If we talk about the stoma structures, they are composed of two bean shaped cells known as guard cells. And the guard cells are dumbbell shaped in the monocot. They are bean shaped in the dicot stomata. Okay, but they are dumbbell shaped in the monocot stomata. When we see the structure of this stomata, the outer walls of guard cells, which are away from the stomatal pore, are thin, whereas the inner walls, which are towards the stomatal pore, are highly thickened. And you can see this is the structure of guard cell. And when you see the outer wall which are thin, you can see these are the outer wall which are thin, and these are the inner wall which are highly thickened, as you can see. So the thick and inelastic wall are shown on the inner side. This is the diagrammatic representation which is given in NCRT. If you see in NCRT, you will see that these are the guard cells which are kidney shaped or bean shaped. And this guard cell, if you talk, they are kidney shaped or bean shaped. And this guard cell have chloroplast. Okay, chloroplast are present, and guard cell have rubisco as well. As important points I will make you to note down in the last part. So here they have chloroplast, but they do not have a rubisco, so they do not perform photosynthesis. In spite of presence of chloroplast, guard cells are unable to perform photosynthesis. And the pore which is found in the center, it is known as stomatal pore or stomatal aperture. Whereas the specialized cells which is present in surrounding to the guard cell are known as subsidiary cells. And on the outside of subsidiary cells, you will see the presence of epidermal cell. So this is the dicot stomata, whereas this is the monocot stomata. Stoma, we can say. In monocot stomata, you can see these are the dumbbell-shaped guard cells. And in dumbbell-shaped guard cell, you will see that inner and outer both wall are thickened. And these are the subsidiary cells. And the surrounding epidermal cell are seen here. Like this dumbbell-shaped guard cell are found in monocot stomata. Now, the guard cells possess a chloroplast and regulate the opening and closing of stomata. But be remember, this guard cell obviously control the closing and opening of stomata. Now, these guard cells, along with subsidiary cells and stomatal pore, stomatal aperture means what? Stomatal aperture is nothing but stomatal pore, then guard cell, and then subsidiary cells together constitute a stomatal apparatus. Together, they are going to be called as a stomatal apparatus. Now, one of the important points that we need to note down here that sometimes few epidermal cells are present in the vicinity of guard cells and become specialized in their shape and size, and they are known as subsidiary cells. So subsidiary cells are nothing but which are present surrounding to the guard cell, and they are somewhat specialized cells. So here you can see these particular points here, that this is the guard cells, okay, and this is the particular opening and closing mechanism. This no need to go this much in detail, but dear student, you should remember that the guard cells have chloroplast, but they do not perform photosynthesis because because rubisco enzyme is absent in chloroplast of guard cells. And when we talk about subsidiary cells, in subsidiary cells, chloroplast itself are absent. So subsidiary cells are not going to perform photosynthesis, as well as guard cells are also not going to perform photosynthesis. So what will happen? They will import the water from the surrounding cells. What they are particularly importing water, particularly food from the surrounding cells, but they are not able to synthesize. So the epidermis bears, this is the what about the stomata. Okay, I hope you understood about this particular stomata very well. So after discussing this epidermis and epidermal structure, this is stomata, what important point we need to note down is that when we talk about epidermis, epidermis is usually single layered, but it is found multi-layered in Nerium, ficus, and begonia like plants. Okay. Apart from this, what next you need to note down is that cuticle is obviously absent in root, but cuticle is also absent in hydrophytes, because in hydrophytes, if cuticle is impermeable to water, if it is present in hydrophytes, it will not allow the absorption as well as loss of water. And in hydrophytes, they are the water loving plants, they are present in water itself. So if the water loss also occur to a large extent, that is not going to affect these hydrophytes. So because of this, we call it them particularly what, cuticle is absent in this particular hydrophytes. Apart from this, if we talk, then In monocot leaf epidermis, that is upper epidermis, bulliform cells are present, and that we will discuss this bulliform cells in the when we will discuss the structure of monocot leaf. But these bulliform cells regulate the rolling and opening or straightening of leaf. Okay, so this is about the monocot leaf epidermis. And after this point, one more important point which we have discussed with respect to the stomata, that is, you should know the this formula, that there is a difference between stomatal apparatus and stomatal aperture. Stomatal apparatus is equal to stomatal aperture stomatal aperture or pore plus stomatal aperture or pore plus we can say guard cell plus we can say subsidiary cells. Okay, so these are going to form a stomatal apparatus. Now after this, many important points in studying the structure of stomata in monocot and dicot, and talking the important points with respect to the epidermis. Now we have to talk about the, we can say, the stomatal appendages or epidermal appendages. Sorry. So this epidermal appendages bears appendages means what? Extension. So here we are going to talk about epidermal appendages, and this epidermal appendages include the root hair and trichomes. Trichomes are stem hair. So the epidermis bear a number of hairs. Okay. Another more important point that this number of hairs are the root hairs, and these root hairs which are present on the root help in absorption of water, minerals from the soil. They are involved in absorption of water and minerals. Usually, root hair are unicellular. Obviously, they are unicellular because they are nothing but the elongation of the epidermal cell. So, epidermal cell is going to elongate and form a root hair. So, root hair are unicellular. So, root hair mainly involved in absorption of water and minerals. Okay. Now, on the stem also you will find some hair-like structure which are known as epidermal hairs or stem hairs, and they are known as trichomes. These trichomes, which are the stem hair, which are found on the shoot system, are usually multicellular. These are usually multicellular. Now, this multicellular stem hair or trichome, if you see, they can be seen as a branched or unbranched structure. They can be soft or stiff. And trichomes mainly help in preventing the water loss due to the transpiration. Trichome prevent the water loss. And they may be secretory, means they can be secretory or non-secretory. Okay, but secretion is one of the important things that can be cause. Sometimes they also catch some of the insects, okay, in their secretion, and due to this, they particularly help to get a nutrition to the some plant also. But this trichome having a such particular features which are present on the stem. Okay, now you should also remember that root epidermis is also known as is also known as epiblema or piliferous layer, or we can say rhizodermis. Okay, so these six things are seen in case of the root epidermis. Now, after discussing this all complete information about the epidermal tissue system, now we are going to talk about the ground tissue system. But before that, let's discuss some of the question. Classification of tissue is done based on the what? The type of cells present, and based on the cells dividing or non-dividing, they are divided into the what we can say meristematic and the permanent tissue. On the basis of structure and location, number of types of tissue system are. So obviously there are three types, that is the epidermal tissue system, ground tissue system, or vascular tissue system. Now outermost covering of whole plant body is obviously it is going to be the epidermis. Cuticle function is it prevents the loss of water, that is what transpiration, because it is impermeable to water. Stomata, if we talk, stomatal apparatus includes, just we have discussed, it includes stomatal aperture, guard cell, and substitutory cells. So according to this, the first option is the correct one. Okay, so now till this we have discussed about the epidermal tissue. Now onwards we are going to discuss the ground tissue system and the vascular tissue system. Now, when we talk about the ground or fundamental, ground is known tissue system is known as fundamental tissue system. Like why? Because majority of the plant body is made up of this ground tissue system, and all tissues except epidermis and epidermal structures and vascular bundles constitute the ground tissue. Okay, so ground tissue comprises of all tissues excepting, means it does not include the tissues which are present in epidermis and vascular bundle, like xylem and phloem are not part of this. Remaining tissues are a part of this ground tissue. So it consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. They are mainly taking part in formation of this ground tissue. As we have discussed, they are simple permanent tissue. And this is the thing in which if we leave the epidermis and vascular bundle, see, this is the stem and we are taking a cross section of stem. When we take a cross section of stem, you will see that this is the multicellular hair on the stem. Okay, this much part is drawn here. Okay, so here you can see this is the epidermis cuticle. If we remove this epidermis part, this is not going to be part of ground tissue system. And same way, the phloem, xylem, and this cambium are not going to be the part of what, or we can say xylem parenchyma also are not going to be the part of which ground tissue system. So ground tissue system mainly comprises of hypodermis. Then you will see the cortex region here. Then you will see the endodermis. You will see the pericycle. You will see the medullary rays, and you will see the pith. This all are going to form this ground tissue system. So parenchyma cells are usually present in cortex, pericycle, pith, and medullary rays in the primary stem and root. And this parenchyma, which is present in this all tissue layers, that is the cortex, pericycle, pith, and medullary rays, these all tissue layers are going to combine and form a ground tissue system in stem and root. In leaves, when we will talk, in leaves you will not see the presence of such cortex, pericycle, pith, and medullary rays type of tissues. So in leaves, the ground tissue is mainly consist of or made up of a thin-walled chloroplast containing cell called a mesophyll cells. Okay, so this is the particularly mesophills. Okay, and these mesophills are present here in between the upper and lower epidermis, and they are mainly formed particularly ground tissue of the leaf. So if we talk about this ground tissue system in detail, if go on talking about this ground tissue in detail, so it extends from epidermis up to the center of axis, excluding vascular tissue. So it consists of number one, cortex. In cortex, if we see, then this cortex lies or present between epidermis and pericycle. Okay, and this cortex is further differentiated into three regions, that is hypodermis, general cortex, and endodermis. This hypodermis is collenchymatous, or it is made up of collenchyma cells in dicot stem. And this hypodermis is sclerenchymatous, or made up of sclerenchyma in monocot stem. And this hypodermis provides strength, but it is absent in root and found in stem. The second part of this cortex is general cortex. And this general cortex consists of parenchymatous cells. And this general cortex, if we talk, its main function is storage of food. And the third one is endodermis. This endodermis is the innermost layer of the cortex. See, here, if we are talking, it is the innermost layer of cortex, that is endodermis, which is usually single layered. Hypodermis is few layered, but generally general cortex is of many layers. Okay, and these three combinedly form a cortex. Now, when we talk about endodermis, then this endodermis is present in mainly root and also in stem. When we talk about in root, here in its endodermis cell, suberin deposition is present as Casparian strips on inner and radial walls. Whereas in roots, this thick-walled suberin deposited thick-walled root endodermis cells with the Casparian strips are interrupted by a thin-walled cells. And these thin-walled cells which lack suberin deposition or Caspian strips are known as passage cells. Actually, the suber is impermeable to water. But when this talk about passage cells, they do not have an endodermis or a Caspian strips. So due to this, they can allow the transport of water. Now this endodermis of root we have as watertight dam to check the loss of water, and airtight dam to check the entry of water in xylem. So inward the air entry is prevented, and outside towards periphery towards outer side the water loss is prevented by this endodermis. So this is known as a particularly one. And when we talk about in stem, then in stem this endodermis is single layered and made up of parenchymatous barrel shaped parenchymatous barrel shaped compactly arranged cells. And it is known that all the tissues which are present near to the endodermis are constitute a stele. So all tissues on the inner side of endodermis, such as pericycle, vascular bundle, and pith, constitute stele. And when we talk about all the tissues which are present inner to the endodermis, then we talk about the pericycle, vascular bundle, and pith. But vascular bundle is not a part of this ground tissue system. So if we talk about the pericycle and pith, this pericycle tissue layer, if we talk, it is lies inner to the endodermis, or we can say between the endodermis and vascular tissue. It is mostly single layer. It is mostly single layer, and parenchymatous in root and sclerenchymatous in stem. Now this pericycle cells itself give rise to lateral root, so lateral root are endogenous in origin. They are endogenous in origin. In dicot root, it forms part of vascular cambium and whole of cork cambium. Okay, so this are particularly found with respect to the pericycle. And inner to the pericycle, the centermost region is known as pith. When we talk about pith, this pith, also known as medulla, occupies occupies the central part of occupies the central part of dicot stem and monocot root, and it is mostly made up of parenchyma cells. So this all cortex, which is made up of hypodermis, general cortex, and endodermis, then pericycle and pith, combinedly form this ground tissue system. Now the remaining is the vascular tissue system, a third tissue system. And this vascular tissue system, if we talk, consists of complex tissue, that is xylem and phloem. Okay. Now this phloem and xylem constitute the vascular tissue system. And along with this together, the xylem and phloem constitute a vascular bundle, or they are known as single vascular bundle. And they also may have a presence of cambium, or they cannot have a presence of cambium. So this is the vascular bundle, one vascular bundle. And the cambium may or may not be present. So if we talk about the type of vascular bundle, then first we are going to talk about the open vascular bundle. Whenever the term is used like open vascular bundle, means there is a presence of cambium. There is a presence of cambium in vascular bundle, or we can say between the xylem and phloem. So vascular bundle in which cambium is present between the xylem and phloem, then it is known as open vascular bundle, and it is found in the dicot mainly, because secondary growth occur in dicot, and for secondary growth only there is a need of what, of this particular cambium between the vascular bundles. So such vascular bundles, because of the presence of cambium, have the ability to form a secondary xylem and the secondary phloem tissues. Hence, they are called open vascular bundle. So this is the, suppose, phloem, this is the cambium, this is the xylem. Now, if phloem is present on the outside and cambium is present on the xylem is present on the inner side, then this particular type is also known as conjoint. Okay, this presence of phloem on the outside side and xylem on the inner side, then this type is also known as conjoint type. And this conjoint, and if cambium is present, we call it as what? Conjoint open. But if cambium is absent, then we call it as a closed. So closed vascular bundle are those vascular bundle in which the cambium is absent. So if in the monocot, the vascular bundles do not have a cambium. Means between the phloem and xylem, you will not see the presence of this cambium. So they do not form a secondary tissue, that is a secondary xylem and secondary phloem. And because of this, it is referred as a closed vascular bundle. This closed vascular bundle is found in monocot, and obviously in monocot, secondary growth or wood or secondary xylem formation is not found. So here, suppose this is the phloem, this is the xylem, and here cambium is absent. So when cambium is absent, if we consider this, then it is a conjoined closed type of vascular bundle. Now, apart from this conjoint can be present in the stem and open in the particular conjoint open in the dicot stem and conjoint collateral in the monocot stem. But radial vascular bundle are seen in case of the root. And in this, xylem and phloem within the vascular bundle are arranged in alternate manner or on the different radii. If somebody asks a definition of conjoint, then in that we say that xylem and phloem are present at the same radius. But in radial vascular bundle, the xylem and phloem are present at the different radii. And this arrangement is known as radial vascular bundle. And here cambium is always going to be absent. So they are in always closed condition, we can say. See here you can see this is the radial vascular bundle, and the radial vascular bundle you can see this is the xylem, which is present on the inner side, and this is the phloem that is present on the outer side, and they are present at the different radius and they are present in a separate patches. Then we call it as the radial vascular bundle. Conjoint vascular bundle, as we have discussed, the xylem and phloem are situated at the same radius of the vascular bundle. One more important point that you should remember: this radial vascular bundle found in are present in dicot and monocot root. Now, conjoint vascular bundle, xylem and phloem are situated at the same radius of vascular bundle. Means xylem and phloem are present at the approximately same radius. Such vascular bundle are common in stem and leaves. Okay, stem and leaves. And the conjoint vascular bundle usually have the phloem located on the outer side and the xylem on the inner side. And cambium may be present or cambium may be absent. If cambium is present, then we call a conjoint open, as in the second diagram which is given in the NCERT. If cambium is absent, then we call it as a conjoint closed. Now this conjoint open is seen in dicot stem, but conjoint closed is found to be present in monocot stem as well as dicot and monocot leaf. So if we talk about example of this, then we can say monocot stem, then dicot and monocot leaf. Whereas conjoint open, if we talk, they are present in dicot stem. Now, bicollateral vascular bundle are also present. In the bicollateral vascular bundles, you will see that phloem is present on either side of the xylem, separated by a cambium. Means phloem is present. Phloem is present on the outer side of xylem as well as on the inner side of the xylem. And this type of bicollateral vascular bundle, if you see, you can see present in the cucurbita. And you can see here that here phloem is present on the outer side, this phloem is present on the inner side, the xylem is present in between, and the cambium is present between both these two. So this is known as bicollateral. Now this bicollateral is found in cucurbitaceae, or we can say members of cucurbitaceae, solanaceae, and apocynaceae family. Okay, so this way, this particular vascular bundle are found in bicollateral conditions. Now, after the bicollateral condition, if we talk about one more type, then there is another type which is known as heterocentric concentric vascular bundle. That is the third type which we discuss, that is known as concentric vascular bundle. Now, when we talk about concentric vascular bundle, in concentric vascular bundle, you will see that in this type of vascular bundle, either xylem surround the phloem or phloem surround the xylem. And this concentric vascular bundle are always closed type, means vascular cambium is absent. Now, if we talk about this concentric vascular bundle, then they are of two types. The first type is heterocentric or amphicribal. Heterocentric or amphicribal. And in this vascular bundle, if we talk, heterocentric means xylem is in center and it is completely surrounded by phloem on all sides. So when xylem is completely surrounded by phloem, then it is known as heterocentric or amphicribal condition. It means xylem is present in the center, and we know that for the xylem we to use a term hedron. So this heterocentric condition occur in which the xylem is at the center. And if we talk about such type of vascular bundle on their example, then they are found in fern rhizome. Okay, so this is the particular thing is found in first type. Now second type, if we talk, the second type is amphivessel or leptocentric. And when we talk about this amphivessel or leptocentric, in this type of vascular bundle phloem is completely surrounded by xylem. So if we talk here, then suppose this is the phloem, then on the outer side it is going to be completely surrounded by a xylem. So vessel, leptocentric, lepto means phloem is in the center, or phloem is present in the center of vascular bundle, then it is known as amphivessel or leptocentric. And if we talk about the example of this, then it is seen in the Dracena and Yucca stem of Dracena and Yucca. Okay, so this is the condition which is seen in case of the, we can say, concentric vascular bundle. Okay, so this is the vascular tissue system. So all three tissue system we have discussed, that is the epidermal tissue system, ground tissue system, and vascular tissue system. Now let's discuss some of the questions. That conjoined collateral vascular bundle in position of xylem is. So when we talk about the collateral, then in collateral, the particularly we can say phloem is present on the outer side and the xylem is present on the inner side. So xylem position is between the pith and phloem, between cambium and phloem, between phloem on both sides separated by cambium. Actually, this is the condition for the bicollateral condition. So here they have written collateral, but collateral in this phloem is present on the outer side and the xylem is present on the inner side, then we call it the collateral. But if you modify here question like bicollateral, then this answer is going to be correct, that It is present between phloem on both the sides separated by cambium. Now, vascular system consists of xylem and xylem and phloem are complex tissue. So complex tissue is going to be the correct answer. Same way, which type of vascular bundles do not have cambium? We know that closed type of vascular bundle have an absence of cambium, so secondary growth cannot occur. So the answer is going to be the second one, that is a closed vascular bundle. The xylem and phloem are situated on the same radius in. It is actually on the different radius in the, we can say, a radial condition, but it is on the same radius in the conjoint condition. So conjoint vascular bundle is going to be the correct answer. Phloem is present on the either side of the xylem separated by a cambia, then it is a bicollateral vascular bundle, obviously, just we have discussed. Okay, dear students, so here all the three type of tissue system, that is epidermal, ground, and vascular tissue system are formed. Thank you.